Hi, first of all, my name is Pam Kreeger. I'm honored to be the Washington Historical Society Roots Award winner for 2020. So I first want to thank the Society for giving me this honor, and I also want to thank my nominator, Mrs. Jan Geis, who I've known for a very, very long time. And I really appreciated her thinking of me as a potential candidate for this. I know that there were many excellent candidates this year, so I'm particularly honored. Um, I'm sorry that I'm not able to be there and actually participate in the normal events, but I'm sure you all understand this is a very different year, and I'll try to come back to that at the end of my talk a little bit. I wanted to give, and I'm sorry, of course, you can't see my face because it's hard to get the recordings always to work. So I'm going to try to do this as simple as possible. Um, I'm going to try to give you a little background about who I am and what I've been doing since I left Washington. So I'm not going to focus a lot on me in Washington, but I went to try to include a couple of pictures of the schools I went to because they were very formative on my life. I went to Lincoln Grade School. I don't remember, and junior high, I think at that time. I don't remember exactly what we called it because it wasn't Lincoln Grade School. But looking at the building that I found in the picture, it doesn't look anything like the building I went to. It's clearly been added on to and expanded and probably is much, much more exciting in some places than it was. But it was still a place that I have very fond memories. I remember second grade math tests and how excited I was to race through and be the first one done. I remember some great teachers. I remember in eighth grade, for example, Maureen Hakovich, who's another person I've known for a very long time and stayed in touch with, who encouraged us to think more dynamically, to think about society and race and issues that we're now talking about in 2020, but probably weren't the most common curricula for people to be thinking about in the early 90s. And so I really appreciate the things that I gained there. Had Mr. Bell was my favorite science teacher probably of all time. And even Mr. Adams for math class, I definitely learned a lot from both of the Mr. Adams that I had when I was there. And then I also, obviously, I went to Washington High School like everybody else in Washington, and it also doesn't look like what I remember. Um, and this is actually a picture of me, I think, in French club in high school. So that's pretty much what I looked like most of the time, just kind of slumped over on my desk. But I definitely enjoyed all the classes I had there. I didn't appreciate it at the time, but I had fantastic STEM education. So science, technology, and, well, didn't have engineering, but science, technology, and mathematics. That really prepared me for what I ended up doing. I graduated and thought, okay, I want to go on and I want to be I think at that time my plan was to go on and be a medical doctor. That was sort of my assumption. I liked science. I liked math, but I didn't think that that was where I would naturally end up going. And so I went to college. I went to a school called Valparaiso University in Northwest Indiana. It's probably best known for two things. One is this picture here, which is the University Chapel. And the other would be its basketball team, which actually I believe a Washington high school player went on to play at Valparaiso a few years ago and did very well there. But at my time, when I was there, was the years of Bryce Drew and the national tournament championship. Or we didn't make the championship, but our run to Sweet 16. So great school for me because I got to do a lot of different things. I did my bachelor's in chemistry. I focused in biochemistry. And I picked up three minors. I minored in math, mostly because I had so much AP credit from having done math at Washington High School that I only had to take, I think, 2 classes to get a minor. And then also a minor in history because I had taken so much AP history from Mrs. Luthi, who unfortunately passed away this year, and was a fantastic teacher at the time when I was there. So because of that, I got to continue. It was worthwhile to continue those things and explore them, and have a little, side diversions while I tried to figure out what I wanted to do with my life, which I ended up getting to through a couple of experiences. So I'm showing you here, the title is my first scientific publication. So this is the work that I did as an undergrad researcher at Valpo. And it was an educational study. So we were designing a lab that college students could do. So not the kind of research I do now, but it taught me a lot about how to think about problems and how to troubleshoot and work through stuff. But the other two pictures, one is that's with the girl there is my first chemistry lab partner, and she is a quirky, funny, wonderful person, and really helped me to kind of branch out from my more small town. And she was also from a small town, but just it was interesting for us to like get to explore and think a little bit beyond what we were used to by hanging out with each other. And then the other picture is showing me at a summer internship I did at Fermi National Accelerator in Batavia. And that sort of introduced me to the idea of engineering, and that while I liked chemistry, I didn't necessarily want to be a chemist. And while I liked medicine, I didn't necessarily want to be a medical doctor. And so what I ended up doing through process of a lot of elimination and conversations and changes of majors and things throughout the process, I ended up choosing to go to graduate school because that's the natural choice if you don't know what you want to do next in your life. And also for some careers, it's what you have to do. And I went on to Northwestern and got a PhD in chemical engineering. So still using my chemistry training and my math training, but now bringing in engineering. And so I guess the lesson I would say from that, if any student is listening to this, is don't think that you have to have your life planned out when you leave high school. You probably shouldn't have it planned out because things will change, and that's okay. Try to make logical choices along the way and leave paths open and shut paths when you realize they're not what you want to do, but you're not going to know what you want to do the day you graduate. So at Northwestern, I pursued my PhD under my advisor, Lonnie Shea, and I worked on a really biological problem, even though I was in chemical engineering. And so what we were interested in is this question of if a woman is undergoing cancer treatments, she might lose her fertility because of the damage of the chemotherapy on the ovary. And so we know that we can cryopreserve these really immature follicles early on in the ovary. And we know that if we have the mature follicle, we can get an oocyte or an egg out of it and do IVF and get an embryo and potentially a fetus and then the birth of children. But there's a missing gap there. The process in humans to go from immature follicle to mature follicle is years and very complex biology that we don't fully understand. And so we wanted to try to develop systems that we could do this where if we had cryopreserved tissue, we would be able to help it to mature in vitro or in a dish and then be able to do in vitro fertilization. And so our model, we use the mouse because it's obviously a lot easier to work with mice than it is to get access to women's ovaries. And so the work, this is kind of my favorite picture for my PhD, is showing you can see the structure that sort of looks like my little drawing. That's a mouse follicle. They're about, at that size, 150 to 200 microns in diameter. And we would manually dissect these under a dissecting scope for hours. And then encapsulate them. And you can kind of see that waviness in the picture. And that's what we encapsulated them in. That encapsulation supports them as they grow and they mature and expand. Because otherwise, if they're just these balls of cells sitting on a dish, they go flat and they all fall apart. And we lose lots of connections that are really important to supporting the egg. So that's what I worked on. I tried to understand factors that would support the development, how we could tailor our system to be more useful. And by the end, we were able to do this entire process with mice. And after I left, the group has continued to work and has made some real progress with potential for human. There's a lot of ethical challenges to actually jumping to testing it in humans. So it's that will be the real leap to make. So at the end of your PhD, you get another graduation, of course, and you get the fanciest robes. And I always joke with people that that's the main reason to get a PhD is that you have a Halloween costume for life because you look like you went to Hogwarts. And so this is the Northwestern robe. It's bright purple. And after that, again, I still didn't really know exactly what I was going to do, where I wanted to go. I knew I liked my research, but I wanted to try something a little different. So I ended up doing a postdoc, which is, if you're not familiar with the term, a postdoc is a period after your PhD, but when you're still considered a trainee or somebody who's learning how to do research. You're an expert in some ways, but you're not, fully independent. And so I worked for Doug Laufenberger at MIT. He's a fantastic, wonderful person. And I really enjoyed working with him. And what I started to learn from his lab was how to think about the individual cells. So my PhD work was like collections of cells and how do we maintain connections and keep them to work together. But each of those individual cells can be thought of as having its own network. So if you look at this and you're at all electrically inclined, you'll see this looks like circuit diagrams. And so Doug's lab and a lot of other labs are trying to understand, well, how do these circuit diagrams work when they're in cells? So how is information processed? How is information taken from outside the cell, turned into signals within the cell itself, and then we call that encoding, and then decoded to make a decision. So if, do your cells need to proliferate? Do they need to die, as a defense mechanism against some kind of injury? Do your immune cells, if you're infected by a virus, how do they turn on a response? So those are the kinds of questions you can use an engineer's approach to try to understand within an individual cell. And you do not get a fancy gown at the end of your postdoc. So the only picture I have here is showing a few years ago. I was able to go back with my son now and show him that was my lab bench. And that's where I worked for about 3.5 years trying to figure out how to do these techniques and what I wanted to do next. And the next step ended up being staying in the universities and becoming a professor at the University of Wisconsin-Madison. So I think at this point it's probably important to clarify what a professor does, because my understanding of what a professor does when I was living in Washington was very different than what I actually do today. I associated it with teaching, and that is one of my primary roles. So I do teach classes, undergraduates and graduate students. However, I have two other roles. One is called service, which is just sort of a catch-all for any kind of committee work. And so some of the committee work that I'm involved in would be diversity work, so trying to increase representation of women and underrepresented minorities in STEM. It's a really difficult problem, and you need a lot of people working on it. So that's one of the activities I work in. Another one is I'm a leader in our cancer center, so I help to try to bring other cancer researchers together. I help to write grants to fund the center so that we have the resources we need to do our work. And the last one, which is most of my time, is I'm a research advisor. So I run a lab. The picture on the right is me day one as an assistant professor in my lab. I literally got an empty room. This picture was taken after we had brought 2 tables into it. It was empty when I got there. And so I started from scratch and, built up my lab. And we work on a lot of different questions, but most of our work is in ovarian cancer. So I want to talk a little bit about some of that work. I will try not to go into my normal science talk because I recognize that's not really helpful if you haven't, been used to hearing those kind of talks. So a little background, of course. Ovarian cancer, like all cancers, is a very concerning disease. The lifetime risk is about 1 in 70 women. Women who are diagnosed with ovarian cancer have a relatively poor survival rate for cancers. Their 5-year survival is about 40%. This has not improved over the last few decades in any significant level. Part of the reason it hasn't improved is that unlike other cancers that we've developed early screens for, most women are diagnosed after the disease has metastasized. And even stranger, we didn't really understand where the disease starts until a few years ago. So we actually think now that ovarian cancer initiates in the fallopian tube, which is the tube that connects the ovary to the uterus, so that when an oocyte, like my PhD work, ovulates, it is transferred down that fallopian tube, potentially fertilized, and then would implant in the uterus or not. We now think that the cells that are on the end of the fallopian tube are in proximity to the ovary, and that ovulation or other injuries are causing these cells to have some sort of stress response. Potentially, that's leading to the beginning of the tumor. And we don't really understand how it metastasizes, but it does appear to go from the fallopian tube to the ovary and then throughout the abdominal cavity. The standard therapy is what's called surgical debulking, which means that we remove as much of the tumor as we can without killing the patient. And I know that that sounds terrible, but that is literally the best we can do when you have advanced metastatic disease, is to try to remove it. But there will be times where the tumor is impacting something such as your bladder or an intestine that you can't do the surgical removal. And even if we think we get it all, and by we, I mean my collaborators who are actual medical doctors, the follow-up treatment is chemotherapy for pretty much every woman. Most women actually do respond pretty well to this. Their surgical outcome is the most predictive of your overall outcome. So if the surgeon feels they were able to remove everything, you tend to have a better outcome. So I always say an important take-home, if nothing else from my talk, is that this is a surgery meant for specialists. It is meant for typically research hospitals and not local hospitals, because it's a very complicated surgery, and we want to do an aggressive surgery to improve patient outcome. But again, most women respond pretty well initially. Eventually, though, the disease will recur and chemotherapy becomes ineffective. So, big complicated problem. How could an engineer help? So a couple of things that engineers do, and I am fortunate to work with many engineers at UW that do this kind of work. We have a lot of tools that we can bring that we've developed for other applications, but they work in biology too. So for example, imaging. I'm showing you here some imaging we did in an ovary, and that white that you're seeing are collagen fibers. And we're looking at the patterns that these fibers make. And we can extract patterns and look and see how those change during disease, and then model that using other methods. The microfluidics. So microfluidics is a really cool tool where you can develop systems that you can culture cells in complex patterns or complex geometries, but doing it outside the body, and then look at the interactions between those cells. Biomaterials. If we want to recreate the human body, we can't use plastic, we can't use glass, and that is our standard materials that most biology are done on. So biomaterials give us a way to recreate some of that, the natural complexity of what the cells see, such as those fibers you're seeing in the image. And then, of course, there's systems biology, the work I talked about from my postdoc, where we can try to understand within the signaling network or between the cells, what is the network of interactions. And I think while these are specific tools that we use, there's an additional benefit of coming at this as an engineer, is that you've naturally had what we call interdisciplinary training. So sort of like in my bachelor's degree, where I had more than one focus. A lot of engineers have more than one strength. They have a chemistry and math, or biology and physics, or some other combination that they're bringing to their work. And those can give you some really unique perspectives. Additionally, engineering in the curriculum in particular tends to be very collaborative. If nothing else, I remember sort of joking as a student that that's how you survive as an engineering student, is you learn to work with each other, because the problems are really hard and you can't solve them all on your own. So what worked for you in high school to go home and do your homework in your room by yourself, or unfortunately, now taking your class in your room by yourself, isn't the most effective way to learn how to handle really complex problems. Being able to bounce ideas off of other people is really important. And so those are, I think, natural skills that engineers bring. So what we are doing specifically, we want to be able to model ovarian cancer metastasis. So again, metastatic cancer means that the tumor has already spread beyond the initial organ. So for example, in breast cancer, we know that many patients will have metastatic disease in their lung, brain, bone, liver. And what we have found through a lot of research is this happens by tumor cells escaping into either blood vessels or into the lymph nodes and then moving through those systems to those distant sites. Ovarian cancer is a little different in that the fallopian tube and the ovary are exposed to the peritoneal fluid, which connects and flows around all of the organs in the abdominal cavity. And so what seems to happen is that the cells can actually detach into this fluid, flow through it, and then reattach and initiate metastatic tumors in these other sites. And so the question is really a different question than if I was studying metastasis in breast cancer. It's a very unusual form, and it probably can occur quite quickly once it's started. So slowing the spread may help to improve patient outcomes. To develop our model, we need to drill down a little bit to understand what are the pieces that would be included. So I'm showing you here the blue are the tumor cells. Of course, I'm only modeling at this point the attachment. I'm curious, how do they reattach? Because if I can prevent that, then I wouldn't have new tumors to treat. So I need to model that. So I need to have tumor cells that are in liquid and that they have the ability to potentially settle and attach. What they would actually attach to is not, again, glass or plastic. It's going to be cells that are lining the outside of the other organs. And those cells are called mesothelial cells. They are sort of naturally, relatively non-adhesive cell. So that's a really big question, is how can they attach? These cells aren't supposed to be, super sticky per se. Once they attach, they then do break through and they interact with the underlying tissue. So that's shown here as this gray extracellular matrix. And so that's one part of the system, but we thought, well, there might be other parts that we would want to include. And the one we landed on was the macrophage. Macrophages are part of our immune system. They kind of exist on this spectrum of either being very inflammatory, so fighting, for example, an infection that you have, or anti-inflammatory, so promoting growth and healing, like if you have a wound. And in cancer, macrophages tend to take on that promoting growth and healing behavior. So they actually end up being supportive of cancers rather than fighting them. And so we were curious, could they also be taking on a role that's helping the tumor cells to attach? So to model this, we developed a new system. We looked at some of the existing systems. We tried to develop some microfluidic systems that ended up being just a little more complicated than what it needed to be. So I'm not going to try to go through every detail, but what we've developed is this mold system where at the end we get these oval rings that you can see at the bottom, and they have 4 little pillars on them, and I'll come back to the pillars. They're actually kind of important. So what we do is we take these rings and we put them in our culture dish, and we can put some extracellular matrix that the mesothelial cells then are cultured on top of. So they're there, and they are ready to potentially have tumor cells attached to them. But we want to add the macrophages. Macrophages, you can get them from human blood, and that's what we do from donors, but they don't exist in that tumor type. We need to make that. And so what we do is separately, we take the monocytes out of the blood, we differentiate them to macrophages. They then stick to this glass so that they're really stuck there. But they're now going to behave like our tumor type macrophages. And so to set up actual co-culture, what we simply do is flip that cover slip, that piece of glass over, so that the macrophages and the mesothelial cells are now encased in some ways, and there's liquid between them. And now I'm going to come back to those pillars and why they are important. They prevent the cover slip from falling off as you simply, you're walking around the lab to do the next step of the experiment. These are two separate pieces, so that they're a way to keep everything held together. They're really a very simplistic approach, and it worked really well, so we've stuck with it. The other thing that's kind of key is the macrophages are on a square shape, and the ring is obviously an oval shape, so that leaves a gap, and that gap is how we can then insert the tumor cells into the liquid and then monitor their attachment, or we can sample the liquid to see what's being secreted by the macrophages so that we can try to figure out what's going on. So when we look at this, we have our tumor cells here. I'm going to talk about the picture that has the black background first. So those green cells are actually our mesothelial cells that we've dyed with a green color. And then sitting on top of them, you can see are the tumor cells, and those are red in this case. Now, if we look at the other two pictures that have the gray background, in this type of imaging, the tumor cells are labeled green. And on the left is a condition without macrophages, and you can see that some tumor cells attach. And on the right is a condition with macrophages, and you can see that more attach. And so that really set up the question that we worked on for several years, is the macrophages are helping the tumor cells to attach, and how are they helping them to do that? So we did some screens to try to figure out what's being changed, and it turns out the macrophages are not affecting the tumor cells, they're affecting the mesothelial cells to make them stickier. So we figured out what they were secreting, what the mesothelial cells were expressing, and then how the tumor cells were attaching to it. And so kind of cutting to that, we have this, what we call in biology, a mechanism. So our mechanism is that these alternative or tumor-activated macrophages make a protein, and that protein then activates a receptor on the mesothelial cells. So it's activating that little squiggly CCR5. That then activates some of those downstream pathways like we talked about in the systems biology, and ultimately leads these mesothelial cells to make a protein they don't normally make. And that protein is this P-selectin, which sits, when they produce it, it is now expressed on their cell surface membrane, and the tumor cell has a ligand to attach to that. It's actually a receptor. P-selectin is the ligand. But they have an interaction where they can attach, and that allows the tumor cells to now attach where they wouldn't have been able to before, because the tumor cells always had the CD24, but the mesothelial cells didn't always have the P-selectin unless the macrophages were there. So what's really exciting at this point, we started looking in the literature because we didn't know anything about these proteins. These were not things I had ever planned to study. And we looked and we started to understand that CCR5 and P-selectin already could be targeted. So there are clinical trials for some antibodies against P-selectin. They were developed not for cancer, but we're hoping to see that if they could be effective for cancer patients. They're ongoing for their other purposes. So we know that they've had good safety results, but we don't know how effective they are. But even more exciting potentially is that CCR5 is a very well-studied protein, has a drug against it, Marivirac, because it's the co-receptor for HIV. So this drug was developed to help prevent infection in HIV and to prevent, to give patients better outcomes. And so we know that patients can take this drug for very long periods of time. We know that these patients often, at least in the early trials, were starting it when they were very sick and they were able to tolerate it, which is more likely what a cancer patient would be comparable to. So we have some reason to believe that this drug could be used in this new population. So our next steps, and what I was hoping to have been doing for the past 6 months, was to test these ideas and test them in vitro. So again, in our systems, we've done some of that and it's all looked very good. But to start testing them in an animal model, because that is really the next step, to see if it actually in the long term slows down the cancers. And then ultimately, we would want to work with our clinical collaborators to see if it does work in the animal model, does it work in patients? And I'm really excited about that work. And then COVID. So I did want to bring this in because this is 2020, and I don't think we can have any conversation in 2020 without bringing up COVID. This is the building I work in. So I actually work in not the first tower, the one in the foreground. I work in the tower that's in the background. These are connected to the university hospital. So on March 15th, I believe it was, we kind of knew this might happen. And I got a text from my department chair saying, if you want to go in and shut your lab down in a controlled manner, you need to go tonight. It was a Sunday afternoon. He knew that our dean was going to close research for all engineers. And I don't work in an engineering building, but I do have to respond to the College of Engineering when they tell me that my students aren't allowed to work anymore in my building because they don't consider it safe. So I went in on Sunday with my lab, and we stopped lots of experiments mid-course. We stopped animal trials mid-course. That is work we will have to recreate and redo, and it's very painful to think about the time and the money lost. But at that time, we had no idea how many people had COVID in Madison. We had no way of testing, and we had no idea of how to slow spread. And if research labs are very tight spaces, so it would have spread amongst people if we had not taken action. I remember one of my students saying, okay, you think we're going to be back in a couple of weeks? And I looked at it, I was like, I wouldn't shut the whole lab down if it was just 2 weeks. We would just act like it was vacation. And I was like, I'll be happy if we're back in 3 months. And he was very shocked by that. But that was based on, I based my 3 months on Wuhan. It had taken about 3 months to get things under control. So I was aware that was potentially a best case scenario. In June, we were able to start going back in because by then we understood that masks and some distancing and limiting the number of people could really limit the amount of spread. So at this point, my lab, which is 7 full-time people, I can have at most 3 in at a time. They have to wear masks full day. They can't work together. So again, that's really challenging for research. They can't train people because you can't be within 6 feet of somebody. So it's very hard to get the new people started. So it's really slowed everybody down. And even the people who are doing COVID research, like they were able to continue doing research, but they have all these restrictions. So it's not our normal situation. But things have gone well. I mean, none of my people have gotten sick, and that's very important to me. They're able to restart their projects. We will see how long it takes us to catch back up. I had hoped to get a ton of writing caught up, and of course, that's not possible because I have 2 children, and so COVID has also meant that I've been a full-time educator, mom, worker, like many other working parents. So anyway, but that's kind of where we were at over the summer, and I don't know where we'll be by the time you see this video because we've had our undergrad population come back, and we shut the university down when our average daily infection rate was less than 20. I think it was about 15 at that time, and I know we weren't fully testing, so it was probably actually higher. But our average daily positive cases in Dane County right now are between 100 and 200. So I am hopeful that we will not have to shut down, but I don't know. So that's about all I have because I obviously don't have any new information. So they asked, and I think I mentioned this along the way, a little advice. So I think the biggest piece of advice was what I said a little earlier, that you don't have to, as a high school student or younger, know your life plan. It can work itself out over time. I think a lot of the undergrads that I interact with have an expectation that they already know their whole life plan. And then if that starts to change, that can be very disorienting and scary for them. So it's okay. You don't have to have it all figured out. And hopefully that also will help you to remember to take advantage of different opportunities to figure out what you enjoy and what you don't enjoy. Um, to take the extra class or the extra activity that may not be the thing that you think is going to help you get into the best college, but if it's what helps you figure out what you want to do and how you'll, be a happy, productive person, then I think that's a much better use of your time than worrying about your resume. I think the only other, piece of advice, this may seem strange, is that I remember always hearing that high school was would be the best years of my life. And maybe for some people that is the case. It wasn't for me. I had great teachers. I had some really good friends, but I would not go back to high school ever. And that's okay too, because I found, I eventually found a niche of other nerds like me, and that we have a ton of fun and we're doing things that excite us every day. And so if you've found your niche of people and you're really happy, congratulations, and that's great. And if you haven't, you can find it somewhere else. And so don't worry. It will work itself out. So again, thank you for this honor, and hopefully I will be back in Washington sometime soon. It's been quite a while, actually, mostly because of COVID. And it's always fun to see how the town is growing and changing. So thank you.